Hello, my dear students. Welcome to my class. I, Dr. Swati Pandya, welcomes you. So, students, today we will start next topic that will be fertilization and implantation. Two words are separate. So, let us understand the meaning of these two words. First is fertilization. Fertilization we have understood earlier. It is actually the fusion of male and female gametes. Here also, during copulation, the semen that is released by penis into the vagina. And this process is known as insemination. These motile sperms, which are released from the male reproductive part that swim rapidly and passes through the cervix, enter into the uterus, and finally reach up to the junction of isthmus and ampulla, which is also known as ampullary isthmic junction, that is present at fallopian tube. The ovum released by the ovary is also transported to the ampullary isthmic junction where fertilization takes place. So, the process of fusion of sperm with ovum is known as fertilization. It is very important event, and in last session we have understood that when the ovum that is released during ovulation at 14th day of menstrual cycle, then this ovum that is fertilized by sperm, and when it is fertilized then only the other further events they takes place. And if fertilization doesn't occur, then the ovum that is released as such and corpus luteum that is formed, and the menstrual cycle again repeats. So here we are considering the ovum that is fertilized by a sperm. So now what happens next? Let us discuss. The fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary isthmic junction. This is a very important thing, and with this reason only we can understand that why not all the copulation they leads to fertilization and pregnancy. So synchronization is very important here. The release of sperm and ovum during same time, that is very important. Now during the fertilization, some of the important events they takes place. A sperm when comes in contact with the outer layer which is known as zona pellucida of the ovum, then only it induces changes in the membrane that block the entry of other additional sperms. So only single sperm can enter, and then the physiological changes they occur which not allow any other sperm to enter inside the ovum. The secretions of actually acrosome which help the sperm in enter into the cytoplasm of ovum through this layer. We have understood the main structure of sperm earlier, and the sperm is having three parts. Let us revise it: head, middle piece, and tail. So, at this head region, in the anteriormost tip region, the acrosome is present, which releases hyaluronidase enzyme, which help in entry of sperm into the ovum. Later on, the entry of sperm induces actually the completion of meiotic division of the secondary oocyte. So second meiotic division is also an unequal and result in the formation of second polar body. Earlier we have seen that first polar body has developed. Now the second polar body develops, and it is and the haploid ovum that is also released, that is also present. Soon the haploid nucleus of sperm and that of ovum fuses together to form a diploid zygote. So in this way, a zygote that is formed when the sperm enters. Here in this diagram you can see Firstly, this sperm has entered, it is very much near to this layer, thick layer. It is known as zona pellucida, and pink color layer that is known as previtelline space, and inside it the ovum that is present. These nearby cells, they are the cells of corona radiata, and these are other sperms. So in this way, the other sperms cannot enter because this is first sperm to enter inside the ovum, and then this layer that is changed to fertilization membrane. So certain changes they occur, and as soon as the sperm nucleus it mixes with the ovum, here the zygote formation takes place. So the meiotic division starts as the zygote moves through the isthmus of oviduct, and this first process is known as cleavage. So further development it starts with this process known as cleavage, and further it forms two celled stage 4, 8, 16 daughter cells because mitosis here takes place. So we all know that through mitosis one cell it give rise to two daughter nuclei, and the two daughter nuclei they are having the same number of chromosome as present in the parent cell. So here zygote is having 2n number of chromosome. So definitely in further cells which are formed, they will also having diploid chromosomes or diploid condition. So here the zygote with the help of cleavage it give rise to two celled structure, then further four celled, 8 and 16 daughter cells. So up till this it is known as blastomeres. So blastomere stage reaches, then the embryo with 8 to 16 blastomeres is called morula. This morula it continues to divide and transform into a blastocyst, and it moves further into the uterus. So all these events actually they are occurring in the fallopian tube, and you can see the same process here in the given picture. This portion that is of ovary, here are the different stages of development of ovum, that is oogenesis. This particular area it is showing the release of ovum. So this is ovum, and near about here, that means if we see the B diagram, A diagram is shown here. This is B diagram which is occurring somewhat here in the fallopian tube or oviduct. So in B diagram you are able to see that how the sperms they are fertilizing here, and this one it has entered. So now the remaining sperms are not able to enter inside, only single that is allowed. Then in C diagram you can see this stage the two cell structure that is developed. So this is converted into zygote, and this is two celled, then after four celled, and this is 8 celled. So this 8 cell structure that is known as morula, and here the E diagram that is showing the morula. Then after morula it travels towards uterus. So this portion is of uterus, and now after the further development you can see here the more cells they have developed, and then the blastocyst has developed, and totally the structure that has been changed, and this particular blastocyst condition, now you can see this blastocyst, now it will get attached to the endometrium of the uterine wall, which is the innermost layer. So this blastocyst condition that will attach to the uterine wall, this process is known as implantation. So let us see here. The blastomeres in the blastocyst are arranged into outer layer called trophoblast. So this is trophoblast, and an inner group of cell attached to trophoblast is called inner cell mass. So this is inner cell mass. This is inner cell mass and this is trophoblast layer. The trophoblast layer then gets attached into the endometrium and the inner cell mass gets differentiated as the embryo. So, in this condition, here it is known as embryo, and here this process is implantation of embryo. So, after attachment, the uterine cell divides rapidly and cover the blastocyst because it has to hold it tightly. So, as a result, the blastocyst becomes embedded in the endometrium of the uterus. So, this is implantation, and implantation leads to pregnancy. So, this is the confirmation of pregnancy. So, in this way, you can see the stages, and this diagram is not important to draw, but diagram based questions you are supposed to learn. So, which event is known as what, which type of process is going on, which part is this, what are the labels. So, all these things you should remember. So, students, we have seen how the fertilization takes place, how the implantation occurs, and these two process, the long answer type question, they can be asked on the basis of this topic. So this is an important topic, so please do revise it and complete all the diagrams properly. Only the ovum which is surrounded by sperms, that diagram, that is important to draw, and do the work properly. If you are having any doubt, you can ask me anytime, and this will help you in the further unit test. So today we are ending here our session. Till then, take care. Goodbye, students.